DNA microarray technology was invented by Patrick O'Brien and his colleagues. This technology was invented to study the difference in gene expression between normal and cancerous cells. The basic principle of microarray is based on the binding of complementary base pair. For example, adenine binds to thymine and cytosine binds to guanine. The binding of extracted mRNA from the sample toward the single-stranded DNA on the glass slide is also known as hybridization. The extracted mRNA is labeled with fluorescent tag to identify and determine the quantity of hybridization. Standard operation procedures. The mRNA is extracted from both the healthy cells and cancerous cells. The mRNA will then be converted into a more stable form called cDNA and subsequently labeled with red fluorescent dye for cancerous cell and green fluorescent dye for the normal cell. Both the cDNA are then mixed together and loaded onto the microarray glass slide. Each spot on the slide contains known single-stranded DNA of specific gene, and complementary binding of cDNA toward the single-stranded DNA indicate the expression of the gene, while the remaining unbound cDNA will be washed away from the microarray. The microarray will then be passed through a laser beam to identify the target DNA pieces through the emission of fluorescent light. Applications of microarray. DNA microarrays can be used to determine gene expression levels. RNA will be extracted from the desired cells and converted to cDNA or T7 RNA promoter-tailed cDNA. It is then converted to cRNA through the Eberwein amplification process. Antibiotic treatment. Bacteria has become more resistant to the antibiotics and subsequently caused more disease outbreak in the modern era. The conventional method of producing antibiotic was to culture large amount of bacteria to determine the effectiveness of antibiotic. However, certain bacteria such as actinomyces are difficult to culture in large amounts. Thus, DNA microarray analysis can be done by diagnosis with a small quantity of DNA extracted from bacteria. This is because bacterial DNA can last longer than the bacteria, and the bacterial cultures are not necessary. Microarray can also be used in cancer treatment. Tumor development involves the alteration of hundreds of cells and genes. Therefore, researchers can use microarray to compare a cancer patient and a normal patient by looking at different patterns of gene expression levels. There are a few types of microarray in current practices. Based on the type of probes, microarray is divided into several types, which includes DNA microarray, protein microarray, peptide microarray, and antibody microarrays. DNA microarray is also known as gene chip, DNA chip, or biochip. It either measures DNA or uses DNA as a part of its detection system. 4 types of DNA microarrays are cDNA, BAC, oligodNA, and SNP microarrays. Protein microarrays act as a platform for characterization of hundreds of thousands of proteins in a highly parallel way. Protein microarrays is of 3 types, and these are analytical protein microarrays, functional protein microarrays, and reverse phase protein microarrays. Peptide microarrays are used for the detailed analysis or optimization of protein-protein interactions. It helps in antibody recognition by screening proteomes. Next, antibody microarrays are protein-specific microarrays that contain a collection of antibodies placed on a microscope slide to detect antigens. Thank you.